All right, so we're talking about the small population paradigm, and that refers to those largely stochastic factors that can result in the extinction of small populations. Now, one of those factors which we've talked about extensively in this class and in the previous video, we talked about demographic stochasticity as a key process that is stochastic and that can affect small populations much more dramatically than large populations. We also got into the concept of genetic drift, which is really just the genetic consequences of demographic stochasticity. And so we went through an example of that. And now I want to talk about another process that affects small populations much more than large populations. Now, first of all, let's look at this figure in more detail. Now, this is a particular example using the golden lion tamarin, but we see this type of result playing out in real systems around the world, where you see that, and this is looking at survivorship. So we're just kind of following a cohort of wild golden lion tamarins over time. So we're looking at a survivorship curve essentially, where you see how the total percent surviving kind of drops off over time. And we see that we have two different populations that we're looking at in this case. One non-inbred population, and we'll describe what I mean in a minute, and one non-inbred population. And we see that the survivorship, there's always a greater percentage of those non-inbred individuals remaining than those inbred individuals. And the inbred individuals, you can see it drops off really dramatically right at the beginning. And so that's saying that especially in early life, the survivorship or the survival rate in the early stages of life is far lower for those inbred individuals than the non-inbred individuals. And so we see this is an example with golden lion tamarins, but we see this all over the place. This process plays out. So what is inbreeding, and why does it particularly influence small populations? Let's look at that. So most diploid populations, and diploid populations just means that individuals have two copies of each gene variant or allele. And that this basically includes everything that we consider wildlife traditionally, everything, birds, mammals, fish, basically all multicellular life that is not plants are diploid populations. And so in diploid populations and polyploid populations too, you can have some bad gene variants around, and I put quotes around that. These are often called deleterious alleles or deleterious gene variants, and these deleterious alleles are floating around in all populations. We all have them basically. They're in our gene pool, deleterious alleles, and they don't usually cause problems because, in general, these deleterious alleles are also recessive. That is, in order for the bad effect to take place from these deleterious alleles, you have to have two copies of that allele. If you have just one copy, and again, we're all carrying two copies of every single gene, if you only have one copy of the bad allele, it doesn't matter. You have to have two copies for a bad effect to take place, for your potential, your reproduction potential to be lower, or whatever it is, whatever bad effect that causes. You have to have two copies. And since these alleles are rare and you have random mating populations, it's just extremely unlikely that you get two copies of the same bad allele, or that you transmit two copies of the same bad allele to your offspring, unless your two parents are close relatives. If your two parents are close relatives, both of them have a relatively high probability of inheriting the same bad gene variant from a single common ancestor. That is, if your parents are related, they have a common ancestor somewhere, and it's possible if that common ancestor probably carried a bunch of deleterious alleles, it's possible that because your two parents share that common ancestor, that they might get the same bad, or that you might get the same bad allele that common ancestor shared. And so it's much more likely, you have a much higher probability of inheriting one of those bad gene variants or deleterious alleles if your parents have a common ancestor. And that is what is called inbreeding. That's what we mean by inbreeding. It means that you have a much higher likelihood of inheriting deleterious alleles from both parents, and that causes you to have a higher chance of having two copies of that same bad allele, and therefore actually experiencing the bad effects of a recessive deleterious allele. All right? So clearly, why is it more likely for your two parents to be close relatives if you're living in a small population? Well, you just don't have that many choices. The parents, the pool of possible parents isn't that big, and so it's quite likely in some cases that your parents may be closely related if the population has been really small for multiple generations. It's just the chance of that is just much, much higher. Now, some species have strong mechanisms to avoid inbreeding. They kind of know that inbreeding, in some way, evolutionarily, they know that inbreeding's bad, and they kind of avoid mating with close relatives. But sometimes, if a population is small enough, you really don't have much choice, and that causes problems. And humans, we do have a choice about there's a large enough population, right, that we don't have to engage in inbreeding. But in some cases, there have been pressures to inbreed, and that the classic example there is royalty in Europe. And some royal families, there was intense pressure to marry a close relative just due to political concerns. Now, this is Charles II of Spain. I just want to point out this as kind of a classic example of inbreeding from world history. And if we look at the Wikipedia page for Charles II of Spain, I just want to point out one paragraph here. Now, he's a member of the Habsburg dynasty. The Habsburgs achieved prominence partially due to advantageous marriages, and protected their holdings by frequent intermarriage between the Spanish and Austrian branches. So Philip and Mariana were his parents, and they were also uncle and niece. So they're pretty close relatives, making Charles their great-nephew and first cousin, respectively, as well as their son. So all 8 of his great-grandparents were descendants of the same 2 people, and that causes problems. That's what we mean by inbreeding, because you can inherit deleterious recessive alleles from common ancestors like Joanna and Philip I of Castile in this case. All right, so enough of that. This isn't a history class, but I just think it's a good example that illustrates inbreeding. Now, those are the processes that really disproportionately affect small populations, and they're stochastic processes. So demographic stochasticity, genetic drift, and inbreeding. Those are the essential population processes that can really affect small populations and ultimately lead to extinction. And this concept is illustrated with the Extinction Vortex, which is a central concept of conservation biology. So I'll just hop over to PowerPoint to show this example. All right, so we can imagine that a population becomes small originally because of processes like habitat loss, potentially epidemic disease, over-harvest, invasive species taking over a landscape. So these kind of processes can ultimately produce small, fragmented, isolated populations. So, and we'll talk more about this as part of the declining population paradigm lecture. But the key here is once the population starts becoming small, fragmented, isolated, we can start to see loss of genetic diversity. By loss of genetic diversity, we mean genetic drift and inbreeding. So these processes become more likely, and potentially that's going to reduce the vital rates, because if you start expressing some of those deleterious recessive alleles, the vital rates, the reproductive rates, the mortality rate's going to go up, reproductive rate's going to go down, which leads to reduced survival, reduced reproduction, which reduces population size, right? And smaller population size makes it more vulnerable to demographic stochasticity, potentially more vulnerable to even, if the range contracts of your population, that might make it more vulnerable to a random stochastic environmental event, like a flood that affects the entire area that the population occupies. So reduced population size leads to increased chance of stochastic events causing extinction. Smaller population size leads to increased inbreeding and loss of genetic diversity through genetic drift, which will potentially lead to reduced ability of the population to withstand environmental events like drought, reduce survival and reproduction. All of this may reduce population size further and make it more vulnerable to stochastic processes like demographic stochasticity, making it more, with a small population, inbreeding becomes a bigger issue, so on and so forth. And the key here is that this process is called the extinction vortex, where a small population is kind of like stuck in this vortex. As smaller the population gets, the more vulnerable it gets, the more inbreeding, the lower the vital rates, the smaller the population gets, so on and so forth. It's this vicious negative cycle that we call the extinction vortex. And even more so, if we take these processes that originally caused the population to become small, like habitat loss, overhunting, invasive species, and we get rid of all those processes, we mitigate them, we get rid of them so that they're no longer affecting the population, it doesn't matter, because the population is already so small that it's embroiled in this extinction vortex and it cannot get out. And that is the tragedy of the extinction vortex. So we just want to, as conservation biologists, we want to avoid populations getting into the extinction vortex, because once you're there, it's really hard to get back out. All right, so that's the extinction vortex. And in general, again, extinction, the final extinction for species is often caused by stochastic processes just because of this extinction vortex. But just remember that stochastic processes are not likely to be the reason the population got small in the first place. And we'll talk more about that in the declining population lecture. Now, one more really critical concept of the small population paradigm is the minimum viable population, or MVP. So this is a really applied concept, and this is an applied population ecology class, so it's a really important concept. As wildlife managers or biologists who work with wildlife managers, we often want to know, how small does a population have to be before we have to start worrying about things like the extinction vortex, like genetic erosion and inbreeding. So this is a question, it's a practical question that we see all the time when we work with populations, especially populations in conservation concern. So we talked about allele effects earlier, so you may see a parallel here with our discussion of allele effects, because in allele effect models with a logistic growth model with an allele effect, we saw where a deterministic population model could have a minimum threshold. So below that critical threshold, the population's doomed to extinction, essentially. And above the threshold, the population would eventually increase until carrying capacity. So there's this critical threshold, and that's kind of like a minimum viable population. But the key difference here is that the minimum viable population concept is really an applied concept, and it comes directly from conservation biology, which is a very applied field, and we deal with, as conservation biologists, we deal with real world systems which are messy, variable, and uncertain, and therefore the MVP, or minimum viable population concept, is usually quantified by using stochastic population models, because we have so much uncertainty. We have to embrace that uncertainty, and we do that by using stochastic population models. And it's really only those models that can tell us what the minimum viable population really is. Now, there's a couple definitions of minimum viable population, but in my opinion, the most satisfying one really is that population size above which the extinction vortex can be safely averted. Because really, as conservation biologists, we just want to make sure that the population that we're focusing on doesn't get so small that it starts to fall into that insidious extinction vortex. However, that definition isn't really quantifiable in many cases because it relies on inbreeding, which is not, it's tough to quantify for a lot of species unless we have lots of data on their genetics and their parentage, and we can understand how inbreeding affects this population. It's not quantifiable that way because we just don't know. And so oftentimes we have a more practical definition, and that's the definition that we're going to use really in this class, a kind of a working definition of MVP that we can relate directly to our stochastic population models. MVP, minimum viable population, is the smallest abundance that satisfies a specified extinction risk threshold. For example, the smallest initial abundance such that the risk of extinction over 50 years is below 10%. And we're going to work with this concept in lab this week. So you'll get some more hands-on experience with this, but you should know right away that we can use our stochastic population models to quantify extinction risk, because we can run, say, 100 simulations. We can count up the total number of simulations that go extinct, say 7 of them went extinct, 7 out of 100 went extinct. That's a 7% extinction risk. So we can easily quantify extinction risk using our stochastic models and running them multiple times and counting up the number of extinctions, dividing that by the total number of replicates. So we can easily compute extinction risk. All right, that leads us directly to the concept of population viability analysis, which really did originate with the small population paradigm. It's one of the foundational concepts of conservation biology along with the small population paradigm, so it's worth talking about right here. Now, of course, you've all heard this concept before, population viability analysis, PVA. That is how we're describing our final projects in this class, as PVA models. Because PVA really is a synthetic exercise where we take all the information that we know about a species, we put it all together into a model and use the model to understand what the implications of what we know are, and what that means for the future, how likely it is for this population to go extinct, what are the potential management actions that could improve the population, what management scenarios are most effective, what are least effective. So population viability analysis is a way to put all what we know about a species into a model and ask questions. Use the model itself as kind of this test tube, experimental playground to understand what the future might look like and how management might help. So population viability analysis, in its first uses, are often used to model processes involved in the extinction vortex. And this includes those processes that are originally caused the population to become small, like habitat loss, disease, overhunting, invasive species. So really all processes that are involved in population dynamics, demographic stochasticity, catastrophes, environmental stochasticity, harvesting, all these processes, put them all together, and that's your PVA model. Now, we are not going to talk much about inbreeding depression in our models because, again, not much is known about inbreeding depression for a lot of the species that we're focusing on, and it's a little more difficult to incorporate data on genetics. So I'm not asking you directly to model genetics in your final projects unless you really want to, in which case please let me know soon, and I can show you how to do this using a software called Vortex. Which brings up Vortex, which is probably the most widely used population viability analysis software. It's free. It's available from, I think, Chicago Brookfield Zoo, but you can just Google Vortex, and you should find the download if you want to play around with it. Obviously, Vortex gets its name from the Extinction Vortex concept. And one cool thing about it is it does allow explicit modeling of inbreeding and loss of genetic diversity, that is genetic drift, in small populations. So this is the central image for, this is the icon for Vortex. You can see all these species. As kind of an extra challenge, try to identify some of these species, because these are all species that have gone extinct. Oh, sorry. some of these haven't. These are almost extinct. These are the ones that have gone extinct. So you can recognize the dodo here, but there's a couple others. Do you know this one? This is the Tasmanian tiger, or the thylacine. Anybody know what this is? So anyway, this is Vortex. It is it's a good software for PVA, but it's a little bit hard to use, so I would still recommend in your final projects that you use Insight Maker. The final thing I wanted to say here is just bring up this example, and the top hat questions afterwards will be based on this example. So here is the Aruba Island rattlesnake, or the cascabel. It is a critically endangered species, and its case kind of brings up a number of important small population related themes. So here is the island of Aruba. It's a very small island off the coast of Venezuela, and you can see it's just geographically it's super small, and that raises a number of concerns right away. And so that's one thing I'd like you to think about is why is a geographically small area, if the entire global population of a species is confined to a small geographic area, why is that a problem? Why does IUCN Red List treat that as, even if there's a large population within that area, just having a small geographic range itself is one of the criteria that can be used to list a species as critically endangered under the IUCN Red List? Why is that? And we're going to focus on this example more in lab this week. But the questions I'd like you to think about now are, why are island endemic species like Aruba Island rattlesnake so vulnerable to extinction? And there's lots of reasons. So I'm going to make this a discussion, and I'd love to hear a whole bunch of different reasons why island endemic species are critically endangered oftentimes. And the other question is, are species that are confined to small areas like the Aruba rattlesnake, are these species more vulnerable to environmental stochasticity than species that occupy larger geographic ranges? Now, just note that I'm talking about environmental stochasticity here, not demographic stochasticity. Environmental stochasticity, that is changes in habitat quality year to year, the population itself getting lucky and having a much higher reproductive rate one year than another, and so on and so forth. Are species that are confined to small areas more vulnerable to bad years than species that occupy larger geographic ranges, and why? And so I will leave it there, and please answer the top hat questions, and I will see you in lab.